to take one foundation class for you, and that is regarding ethics, attitude, and professionalism. And in the part of ethics, there are something which is regarding how to behave, how to show what? Your courtesy to your teachers, not only teachers, your seniors, respected sirs. There are some decorums. While one teacher enters into the class, while he goes out, so at that time you must show your decorum. This is not for today, this is you have to maintain for always. Okay, I think you will be following? Yeah. There are a lot of new students, more will be coming. And yesterday, it was, last class was yesterday. Yesterday, I couldn't finish, there was some part. So I'll try to finish in this. I am having some time because one class is shifted. After my class, this class, there is biochemistry class. This biochemistry class will be shifted next. And after that, there was physiology SDL. So I will be continuing with physiology SDL. Okay, so I'll try to complete those parts as well as this topic. What are the competency number of this? Hemoglobin. Find it out from your syllabus. Those who are new, they can find it out from net. You have to write in Google CBME 24, CBME 24, NMC. So you will be able to download a document, a long document, and all the syllabus of all subjects are there at the bottom. So you will find that. So for every content of syllabus, one code is provided. So in the routine or while I will be delivering my lecture, so I will mention which topic I am, say for example 2.3. Today my topic is 2.3. Okay, so immediately you will be learning what is the topic and what is the content of this topic. So for you, I am telling the content of this topic is hemoglobin, its classifications, what are the different types of hemoglobins, and what are the variations, normal values, variations, and hemoglobin forms, different type of compounds. So I will also study that. Next, the structure. The structure of hemoglobin, I will be explaining, and then the functions of hemoglobin and biosynthesis of hemoglobin and degradation of, catabolism of, breakdown of, same thing, of hemoglobin. Okay? And the conditions in which hemoglobin level comes below normal. So whenever you will get somebody's hemoglobin level less than normal, so immediately your picture should come that either this or this and this may be there. So we have to correlate with other signs, symptoms, other laboratory values. So this is the topic. What is hemoglobin? Hemoglobin is the red pigment, red colored pigment present in, where it is present? Present in RBC, red blood cells. And this is protein in nature. And this is protein in nature. We classify proteins. We classify proteins into three categories. What are these? Simple protein, conjugated protein, when simple protein remain attached to a non-protein substance. Say glycoprotein. Glyco means carbohydrate. When protein is attached to a carbohydrate, then it is glycoprotein. Lipoprotein. When protein is attached to lipid, it is lipoprotein. And when protein is attached to a metal, it is metalloprotein. When protein is attached to a colored substance, it is chromoprotein in that way. Hello? You next to black shirt. Are you following my class? Are you disturbed by okay. If you feel disturbed there, you can come here. Try to follow what I am telling you. So, hemoglobin is having a metal, so it may be classified as metalloprotein, and hemoglobin also having a color, red color pigment, so it can be termed as chromoprotein. So hemoglobins are chromoprotein or metalloprotein, and it's big in size. Okay, since it is big in size, it cannot come out of the membrane, cell membrane of the RBC. It remains within the cytosol, partly remains attached to the cell membrane. But there are some conditions, but not normal these conditions. The hemoglobin from RBC may come out and can be presented in cytoplasm. Sorry, in plasma. In plasma. This is not a good condition. I had one question yesterday. Today I will be seeking the answers from you. Okay? Now I think you are able to answer today. Okay. Later. The main functions of hemoglobin is what? To carry oxygen from lung to the tissues, to all each and every tissues. Otherwise the cell will die. It cannot, without oxygen, it cannot generate energy. Without energy, cell cannot survive. It cannot perform its physiological functions. So, this is very essential. So, hemoglobin is performing very vital function from carrying oxygen from lung to the tissues. Next, in the tissue, what does cell do with oxygen? With oxygen, it oxidizes food, carbohydrate, fat, protein, it oxidizes, and what I get at the return? Energy. And I get another byproduct for this catabolic process, carbon dioxide. So what the cell will do with this carbon dioxide? If it retains this carbon dioxide, what will happen? It will form carbonic acid, H2CO3, and then it will be dissociated into hydrogen ion plus bicarbonate ion. So because of there will be accumulation of hydrogen ion within the cell will become acidic, and in the acidic medium cell cannot work. So it has to be eliminated from the cell. So from the cell this will be eliminated into the plasma, RBC, or sorry, in other cells, somatic cells, from the cell into the extracellular fluid, from the extracellular fluid or interstitial fluid, it will enter into the blood. Then it will be carried by the hemoglobin to the lung. And from the lung, it will be delivered to the exterior, to the environment through our expiration. Okay? So this is the main function of hemoglobin. Carry oxygen, carry carbon dioxide. Okay. Then another function is transfer, that one. Another function of, since it is a protein, it will be acidic, accumulation of hydrogen ion, the carbon dioxide will bind with water and will form H2CO3. So this will be dissociated, producing hydrogen ion plus bicarbonate. So accumulation of carbon dioxide means accumulation of bicarbonate. The cell will become acidic, environment will become acidic, and in the acidic environment, the cell enzymes cannot work. So they will stop their functions. As a result, the cell will die. So I cannot permit to stay carbon dioxide within the cell. It has to be eliminated. So it is eliminated in the two with the help of the carbon dioxide. So when a person is very sick, in the hospital what is given? Oxygen is given. But what will happen? If there is no hemoglobin, less hemoglobin in the blood, who will carry? Then in that case I have to transfuse blood also. Means through blood I am providing RBC, and this through RBC I am providing hemoglobin. Thus hemoglobin will be carrying the gases, respiratory gases. Another function, while we are studying the functions of plasma protein, every plasma protein fractions had different functions, and one common function was all proteins, either in plasma or in cell, they act as buffer. They act as buffer. So hemoglobin is a protein. It is having globin. Globin is the protein part, simple protein part, and globin is made up of numbers of amino acids. And these amino acids are having amino group and carboxylic group. So this carboxylic group, when blood becomes alkaline, this hydroxylic group dissociates, give rise to hydrogen ion in the medium. So providing hydrogen ion means the cell it is acidic, so it will gradually counter the base alkali and the pH will come down to normal. Any doubt regarding this? No? Or yes? If you have somebody who is having any doubt, please ask. Please tell. No. Either it is protein. I abbreviated protein as PR. As PR is made up of amino acids, and there will be one terminal amino acid there and another terminal acid there. These are the two ends of the peptide chain. Okay? And in between, those who are there in between, they have utilized their amino group as well as carboxylic group. But that person and that lady, their carboxylic group and his amino group is free. So this amino group and this carboxylic group, carboxylic group can behave like this. By donating hydrogen ion. So in this way, this carboxylic group can add hydrogen ion in the situation where base is added to my blood. So it will, in this way, it will counter alkali, excess alkali. Okay. Now you have added acid. You have added acid means hydrogen ion. So what will happen if you add this hydrogen ion, extra hydrogen ion? This will also be taken up by the amino acids. Which amino acids? Which group? Amino group. So this amino group, R is the amino acid. So it is having amino group So this will combine with this and it will form RNase3 plus. Finish. pH is now normal. So in this way, hemoglobin instead of PR, instead of it is HB, hemoglobin, any buffer should have two components. One component is HHB, means this is the hydrogen of this liberated, and another partner is HB negative. Because in globin, same picture, globin chain, at the terminal carboxylic group, amino groups are free. So, same way they will behave. If you add alkali, this will be released. If hydrogen ion is added, this will accept this HB negative and become HSB. Clear? Clear to all of you? Okay. So it is the buffering action of hemoglobin. So this function also hemoglobin does. This transport function actually of gases, respiratory gases, we call it respiratory gases. Why? What is breathing? Why do we inhale and exhale? Why? We inhale to get oxygen from the atmosphere. Okay? And we exhale because we have produced some carbon dioxide which are not needed in my body. These are harmful for the body, so I exhale it into the air. So this is respiration, taking in of oxygen, giving out carbon dioxide is respiration. So these respiratory functions are actually performed by hemoglobin. Its function only to take the oxygen to the cells and whatever you want to do it by. And after that, you will produce something which is not wanted in the cell, so I will bring it back to blank. Each red cell, every cell is having approximately 600 million molecules of hemoglobin. Hemoglobin is the 1/3 of the blood cell. It occupies 1/3 of the blood cell. Synthesis begins where? Hello? From where you get hemoglobin? RBC? From where it comes? RBC? Bone marrow. Okay? There are some progenitor cells. Okay? They will multiply and those multiplied cells will be transformed through various stages. Ultimately, it will produce reticulocyte and then mature erythrocyte. So, in between there is a stage, normoblast. Before that, there is intermediate normoblast. So, hemoglobin biosynthesis starts in bone marrow during the generation of RBC, in which stage? Intermediate normoblastic stage. Okay? And the production of hemoglobin ends when our reticulocyte is formed. When reticulocyte is having before that, these premature RBCs are having nucleus, but in RBC it is gradually diminishing. Nucleus is diminishing. Then in reticulocyte stage, no nucleus. Then RBC is released into the blood without having any nucleus. Hemoglobin has two parts, heme and globin. Heme has, I will explain the structure. Globins are protein, and these proteins are actually, there are 4 peptide chains. This is one peptide chain, that one is another peptide chain, and last two are third and fourth peptide chains. Okay? We, the adult human, we are having 2 alpha chains and 2 beta chains. Alpha chains are having 141 amino acids, and beta chains are having 146 amino acids. 141 amino acids, 142 amino acids. Suppose this is alpha chain. So what I did, now it is alpha chain. What I did, hey, come out. Sit here. Sit here. Now it is not alpha chain. Why? Sequence is changed and it will start malfunctioning. It will not be able to bind oxygen. And abnormality of hemoglobin comes because of this. Change of sequence. Replacement of a particular amino acid by another amino acid. So in this way we will find different forms of chains. In that case, it may be gamma chain. In that case, it may be delta chain, epsilon chain. So, different kinds of chains. Normally, we have, in most of the cases, in adult, we termed it hemoglobin A. We have another variant, A2. Instead of beta chains, alpha chain is common. Instead of beta chains, there is another chain we are coming into. So what is red pigment? Actually, hemoglobin is a conjugated protein responsible for red color of the blood. Deficiency is called anemia. It is known by everybody. And the disadvantage of free hemoglobin. There is a question mark because you will be answering. I put this question mark for you. And among the hemoglobin molecule, globin is 96% and heme is 4%. How much protein, how much hemoglobin you have in your body? Can you assume? Can you predict? Simple. How much? What is the normal amount of hemoglobin in your blood? Suppose it is 15. Suppose it is 15. Hey, where is the unit? You have answered 15, but this sort of answer I don't want. You have to mention what? The unit. He didn't tell me that unit. So, give me answer properly. 15? Milligram or gram? Milligram. 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 Hey, gram. I will be confusing you, but you have to answer the right one. It is gram per liter or per 100 ml. Again, I am confused. Per ml or per 100 ml. So in 1 liter, how much hemoglobin will be there? If 100 ml contain 15 gram, then 1 liter? 150 gram. Then you have 5 liters of blood. 150 into 5? How much? So is that figure wrong? 700 to 900 gram? In your body you can contain how much? This much. Can you calculate it? You calculate it now? Okay, understood. A molecular weight of hemoglobin is 68,000. Hello. If it is released into the blood, plasma, from RBC to plasma, what will happen? Its atomic weight, its molecular weight is 68,000. It will create osmotic pressures like albumin. Albumin is having 66. So it will start, immediately it will start creating osmotic pressure. And we have explained what happens is in case of if osmotic pressure of blood plasma is increased, what happens in capillary fluid exchange? There will be accumulation of extracellular fluid. There will be accumulation of extracellular fluid, means there will be edema. There will be edema. And this excess free hemoglobin will be passing through the kidney, and while passing through the kidney, it will damage the kidney. So, we have to be very alert. The patients, those who are having hemolytic disorder, they will be suffering like this. Viscosity will be changed, osmotic pressure will be changed, there will be anemia, unable to carry oxygen, unable to carry carbon dioxide, lot of problems will be coming. Structure. Hemoglobin, I told there is two parts. One is heme, another is beacon. You missed 1 or 2 classes. 1 class. You like to sit there only, no? I want to know what is the advantage of sitting there. Can you tell me what is the advantage? And then I will ask all the students to sit at the back, if there is any advantage. Yes? No? Then? Okay. So one part is heme, another part is globin. So I told the globin is made up of 4 polypeptide chain. Out of this there are 2 alpha and 2. There are some variations, I will explain the variations. And the number of amino acids present in, and how much of hemoglobin out of the total hemoglobin, how much is the contribution of globins. Most of the part is contributed by globin. Now the heme. Heme is made up of 4 pyrrole rings. It's like pyrrole rings. This is actually C, carbon. Carbon has how many valences? 4 valences. There are 2 more valences. Only here I can find only 2 valences. So one is H. So now it is 3. So another balance is open. So this is another balance is open. This is not C, this is N, nitrogen. This is nitrogen. Spiral ring. Heme is having 4 such spiral rings. So I am arranging these 4 spiral rings. One, two, 3, 4. And these rings are attached to one another by methane bridges. I'll put methane bridge later. So here is nitrogen. Let's see. This is methane bridge. Methane. The 4 valencies are not satisfied. Satisfied? No. So if I put one double bond here, now it's okay. So this is the methane bridge. Here also another methane bridge like this. Here I cannot put double bond because then it will already it is 4. So I can, here it is 3. So I can put a double bond here. Now here also this carbon is also having 4. This carbon is also having 4. One goes to hydrogen. Okay? So in case of this, can you put double bond here? No. Because 1, 2, 3, 4 already satisfied. But here you can put double bond. Okay? Here you can't, you can put double bond here. Okay? Done? Complete? Oh, another bridge is there. C is bridge. 4. Done. Now this is when they are united. Now it is protoporphyrin. And now this all the pyrrole rings have some groups attached to this carbon. These are methane group. Methyl, vinyl, propionic acid, methyl, Propionic acid, methyl. Now it is protoporphyrin. Now this nitrogen of every pyrrole ring, when attached to iron, it will become heme. It will become heme. Iron is having 6 valences if it is in ferrous state. So here is the iron in ferrous state. One valency goes to this nitrogen, another to this one, another to this one, another to this one. 2 valencies left. With one valency, it has to remain bound to globin. It has to bound with globin, but how many globins are there? 4. 4 globins are there. Every chain needs one heme. There are 4 chains in hemoglobin molecule, and every chain must have one heme. So, with another valency, it will bind with, say, alpha 1 chain. Why? What will happen to me? Okay, wait, you will get. Okay? So another heme molecule will come, will connect to this chain, beta chain, 2 beta chains, B1, B2. So B1 needs one, B2 needs one. So one molecule of hemoglobin can bind how many heme? 4. Now see, another bond is left. With this, it can bind oxygen. Now it is oxyhemoglobin. Since the molecule is having 4 heme residues, so one molecule of hemoglobin can bind 4 molecules of oxygen. So this will carry 1 oxygen, this will carry 1 oxygen, this will carry 1 oxygen. Now, can it bind with another molecule? No heme left in this molecule. So it is the saturated form of hemoglobin. Now, this molecule is 100% saturated with oxygen. You went to see a patient, and monitor is attached to the patient, and it is measuring oxygen saturation. The value is 96%. That means the patient is having so many hemoglobin molecules, and all the hemoglobin molecules are saturated with oxygen 96%, not 100%. Means we can say, but if the saturation is 60%, then condition is bad. Isn't it? One molecule of hemoglobin, 1 gram of, you have 14 gram. She is having 15 gram, suppose. She is having, Debulina is having 15 gram per 100 ml. 15 gram. 1 gram of hemoglobin can carry at the maximum 1.34 ml of oxygen. This is the capacity of the hemoglobin. 1 gram maximally, when it is 100% saturated, all the heme are bound to oxygen, then we call it 100% saturated. In such condition, 1 gram of 100% saturated hemoglobin can carry how much? 1.34 milliliter of oxygen. Now, your blood, 100 ml blood is having 15 gram. Debulina is having 15 gram. So, how much oxygen Debulina's 100 ml blood can carry? 15 gram into 1.34. It will be around 20-21 ml. So, our blood carries, when it carries oxygen, it will show, it can carry maximally 20 to 21 ml of oxygen. This is the oxygen carrying capacity of my blood. এরপর তুমি যতই অক্সিজেন অ্যাড করো, no use. Who will carry? This is the capacity of my. In that case, you have to add extra hemoglobin. You have to add transfusion. You have to give a transfusion so that this extra hemoglobin can carry if it is below normal. Otherwise, I will not add, because this adding extra hemoglobin or adding extra RBC, it is having a deleterious effect. We have discussed. It will produce a lot of physiological consequences. Is that clear to you, structure? Any doubt? Clear? Very good. You are not writing.
Why is there bond left in CH?
Which one?
Left one. Stop.
Oh, this one. It will not be there. Oh. Redo? Undo? Yes. This one, no? What? This will not be there. Very good. It's okay now. Intelligent boy. Can I proceed now? See how the peptide chains remain. And one heme part actually remains exposed. Other remains hidden within the molecule. They are not exposed, not at the surface. When one molecule of one heme becomes able to bind oxygen, what happens? The other heme moieties, the other iron part, heme part comes out to the surface. Now they can, so now its affinity for oxygen, initially affinity was less. When one molecule could bind, immediately affinity for oxygen is increased. Oh, I started liking after taking one. Rasagula, it's good. It's very good. Give me more, give me more. It's like that. Functions already. Another function, last one. You may not find it in all the books. What is hemoglobin? Hemoglobin. There are other forms. These are less significant, so I will not waste my time here. You go through the competency, you will find what are the topics, subtopics there under this competency. So you have to learn that, and you have to learn today's portion. So I will be asking you questions. I means there are other teachers will be there. They will be asking you questions, so you will be evaluated there. Formative evaluation. It is written there, formative evaluation. So prepare for that. Breakdown. Old fragile RBC, lysis takes place. It is breakdown of hemoglobins take place. So What happens? Lysis of the RBC. Membrane is ruptured, then hemoglobin comes out, and this hemoglobin, by the enzymes are there, this enzyme will split hemoglobin into heme and globin. Globin part will be separated, all amino acids will be released, so they will be sent back to the circulation. And you need it, take it. You need it, take it.
Circulation in the blood. Circulation
Into the means in the blood, from spleen into the blood, from liver into the blood. What liver will do? It can store, but whenever it is required, it will supply. Then the heme part is converted into biliverdin. After liberation of iron, this iron will be recirculated, reused, recycled. Okay? It will go to the bone marrow through liver. First from spleen or will go to liver. Liver will send it to the bone marrows. Okay? Then biliverdin will be changed into bilirubin. It happens in liver. And this bilirubin, this conversion is taking place in the liver hepatocytes. When these hepatocytes are broken down, lysed by viruses or by some other means, the contents, bilirubin is one of the content. So this bilirubin, along with some other enzymes of other pathways, like LDAs, like SGPT, SGOT, these are the transaminase enzymes. These enzymes, bilirubin, they will be coming out from the liver cells because liver cell is lysed, broken down by the viruses. So this will be coming into the blood. So blood levels of bilirubin will be raised. Your body will be, what color of your body? Will be yellowish. Your mucous membrane will be, that means there is bilirubin accumulation. Means you are suffering jaundice. So, when blood levels of bilirubin...
Back to jaundice part, explain this again. Jaundice part, explain this again.
How jaundice develops? Jaundice develops from the liver. Breakdown of liver cells, we call it hepatocytes. Say some hepatitis virus. Say when it infects liver, so liver cells are damaged. Plasma proteins. If concentration of albumin is hypo, then this can happen. If concentration is more, these are the conditions